Wherever you are in the world, Optica means business. Next Tuesday, September 26, Optica is organizing another global online technology meeting. And this time, we want to discuss transformative impact of biosensing. Using optics-based gene sequencing and cutting-edge biosensors, optical engineers swiftly decoded COVID-19, paving the way for groundbreaking vaccines and treatments. But this is just the beginning. Today, biosensing is key to real-time glucose monitoring for diabetics, swift pathogen detection in food, and more precise cancer marking. At Optica, we want to help corporate members leverage the maximum business opportunities in biosensing. So we have put together a world-class group of people who will share what they need and who can help them. Look who's coming. Benjamin VerSteeg, Executive Vice President of Rockley Photonics. Scheres, Co-Founder of Surfix Diagnostics. Petteri Ushima, CTO of Modulite. Thanosis Manolis, Principal Engineer at biosensor manufacturer Biolum. We welcome back Werner Meintele from Diamond Tech, based in Frankfurt am Main in Germany. Anton Vasiliev from Legend Tech. Jeffrey Owen Katz, co-founder of Sanguis Corporation. Sebastian Hulk, the CEO of Tech5, also coming from New York State. And the fascinating Nadine Walter, co-founder of a specialist imaging technology company, Expex AI, based near Cologne. And Brain Martz, CTO of Atomica, joining us from Santa Barbara, California. We also invite BioStars to share one slide. Our technology scouting team just came across a new Swiss startup that uses near-infrared spectroscopy to measure oxygen levels in brains of premature babies. The company is called Oxiprem, and don't miss this one. We've also asked critical academics like Laura Lechuga from the Spanish National Research Council to join us right now at the Optica Online Industry Meeting. Let's see who else is here. Let me see. I have Bram Bingerling, I have Katrin Boske, I have Clark, I have all of you. Thank you very much for joining this very important meeting. All of us are tired of hearing that photonics has key applications in industrial monitoring, in photonic integrated circuits for datacom. Today, my friends, today we are saving and improving lives. Thank you very much, all of you, for joining an online industry meeting on biosensing. When I said who is in the room, who else is in the room, I want you to know that we are overwhelmed by the interest that this meeting has had in the photonic industry. The companies that registered today for the meeting, we have from optics manufacturers all the way to companies manufacturing MEMS, all the way to microfluidics, fiber optic companies like OFS, like Senko, like Subcom, infrared detectors like Vigo, market intelligence companies like PwC. We had the thin film giants like Viavi or Auxora. We are extremely excited to have the photonic integrated circuit community companies like Fix, like VLC. All these companies have registered for this meeting. And if you want to get in touch with any of the participants today, all you have to do is send me an email and I will be more than happy to make the introduction. You know how this works. We have two sets of companies. We have integrators and early adopters. We're going to show 6-minute presentations about the kind of things that they can do for you and what you can do for them. And then we have one slides from enablers and solution providers who are going to show only one slide about how they can match the interest from integrators and early adopters. But every one of you here in the Zoom room is welcome to unmute the microphone and say at any time what your company does and how you can help each other. We're going to make this meeting as interactive as possible. We started on time. We're going to finish on time. We started at 4 p.m. Central European Time. We'll finish at 6 p.m. Central European Time, which is the same as noon in the Washington, D.C. I also would like to say that this is the biosensing meeting 26th of September. Next meeting we have on the 31st of October. Remember, the last Tuesday of every month, the next meeting is on augmented reality. Augmented reality. All those companies manufacturing micro optics, meta optics, all the companies manufacturing light engines. You have to be there on the 31st of October. But today is all about sensing, and I have a fantastic group of people in front of me. I must say, Laura, I'm a bit nervous because doing the role Laura Lechuga, I don't know what I'm doing talking about biosensing. Thank you so much for joining all the way from Barcelona. And thank you, all of you. Are you all ready? Let's get started. All of you have told us, they challenged us. They said, if you organize a meeting on biosensing, you need to bring Rockley Photonics to the room. And that was a big challenge. We contacted them, and 1 minute after, they said, of course we are there. Benjamin Van Steeg, thank you very much for opening the biosensing Optica industry meeting today. The floor and the attention of everyone goes to the company everyone wants to hear about. Rockley Photonics, the floor and the attention of everyone is yours.
Nice to meet everyone, and I appreciate that. I'm going to have to ask my colleague to go ahead and share the slides as my laptop's having issues this morning. So this is my first opportunity to present to Optica. I plan to talk really about what we're doing in non-invasive optical wearable sensors. And so I'm going to try to do a brief background and then happy to answer questions. So on the next slide, a little bit about Rockley. Everyone is very familiar with Rockley's foundation in silicon photonics. What you may not be aware of is over the past several years, we've acquired and developed expertise sort of across the technology stack. So we have a talented ASIC design team. We also have full integrated product development groups, as well as expertise in the measurement science, including clinical tests and development. On this slide, you can see our Bioptic line of non-invasive wearable sensors. So starting with Bioptics band, we are using novel Rockley designed sensors to add new capability alongside more traditional LED-based sensing. So obviously we also have pulse oximetry, heart rate, but in addition, using our integrated sensors, we're adding hydration and body temperature to our Bioptics band. With cardio, we're actually bringing non-invasive blood pressure, really with an emphasis on being able to provide accurate intra-subject tracking and changes within a user over time. And then with our Pro line, and we'll talk about the extended spectral region, we're expanding the full spectrum there, and that will allow us to bring to the market things like glucose, ethanol, and lactate. And so on the next slide, you can see an example of our Bioptics band where we've integrated our silicon photonics PIC chip alongside more traditional LED sensing. So the PCB inset on the upper right, you can see that includes our proprietary photonic integrated circuit that allows us to do spectral sensing and measure both hydration in the dermis of the skin, as well as the temperature of the water in the dermis. So on the next slide, you can see the heart of and foundation of our technology. And that really is the silicon photonics platform that Rockley has developed over many years. This platform allows us to cover a very broad spectral region. So we have the ability on a single chip to integrate over 100 lasers, all the way down from 1200 nanometers up to 2400 nanometer region. This allows us to make the types of spectral measurements we're going to talk about in a moment. But what's also important is that it's a wafer level technology. So we have the ability to bond and integrate our 3.5 technology at wafer level. We also do electrical in, optical out testing, so we can yield known good die into the downstream processes and maintain high yield. So next slide. So why are we working in that spectral region? Well, when you're looking at measuring compounds in the dermis of a human, this is a unique region that allows you to balance being able to get good chemical information, as well as being able to penetrate light down into the dermis of the skin, several millimeters, where you're wanting to measure these compounds. And you can see here the spectral signatures of several different biomarkers of interest. And then on the right, you can see sort of a cartoon version of a spectrophotometer sensor that you just saw in the wearable. where we're able to illuminate lasers in different digital patterns and then demodulate the signal on the photodiode and extract the spectral information from the skin. So next slide. So the reason why it's important to have this kind of integrated platform is in order to get the signal to noise requirement, the optical resolution, the number of wavelengths, and the broad region, you need to be able to put several milliwatts of power at every wavelength of interest into and out of the dermis. And you also need to be able to do that in a compact platform. So the fact that we can multiplex the light out of a single mode waveguide then enables us to have very compact coupling optics into the rest of the system. And you can see here, the spectrum on the right, this is back one slide, please. This is a human spectrum from the dermis. It's got some little bumps and wiggles because this is the
I think we lost connection with the audio signal of Benjamin.
Can you hear me now?
Yes, we can. Loud and clear. Continue.
Very good. So as I was saying, the plot on the left is a spectrum from one of our sensors. You can see the broad region. The gaps where you don't see information is intentional. And that's because you see on the plot on the right, those bands are really directly under the water absorbance features. And so if I were to put lasers in that region, I would simply be gently warming your skin, excuse me, as opposed to getting information back. So just have a couple more slides left. On the next slide, I just wanted to sort of highlight for the group, we've recently published a white paper. You can follow the link online to get the details of the study. But in addition to the unique spectral-based sensors that we've been talking about, we also are bringing to commercializing a novel blood pressure sensor. And this uses pulse wave analysis. The inset on the left, you can see the comparison between signals from an indwelling arterial line pressure sensor in a clinical study. The blue trace shows our Rockley laser-based sensor measuring non-invasively. And then the green is a concurrent, more standard PPG signal. And as you can see, we're getting a lot of much better signal fidelity, as well as a lot more information content. And that allowed us to generate the results you see here. I'll just touch on a couple highlights. The scatter plot in the middle is showing across population and within subjects, the ability for us to not only measure systolic blood pressure, we also measure diastolic. This happens to show the systolic results to the accuracy required by the ISO spec, but we're also showing intra-subject tracking, and that's important. And on the plot on the right is showing our results against the newly adopted standard, the IEEE standard, which requires that you show good tracking across different blood pressure regions as you move away from a person's sort of calibration point. And so the goal is to get all the blue bars down in the green band. And you can see that we are doing so today in our recent study, with the exception of a couple of the extreme regions where we just don't have enough data in our current study. So we're very excited about these results, and I'm looking forward to getting to present to you again as we bring these to market. So I believe that's my last slide. So happy to take questions.
Thank you very much for being with us today. It is great to have you here. But at the end of the day, what you're doing is offering your silicon photonics expertise to make spectroscopy-based solutions for biosensing. The question here is the entire industry, from potential customers and suppliers are in the room. So what can you do for them, and what can they do for you?
Okay, so we obviously do have unique silicon photonic design capabilities and implementation, and as well as with our fab partners, the ability to manufacture at scale. So one of the things Rockley can offer, of course, is the ability to offer custom silicon photonic solutions, all the way up to folks who are interested in integrated sensors and modules. So we're sort of across the vertical stack. In terms of what we're looking for from the industry, you can imagine that these integrated products require not only the silicon photonics technology, but also expertise and continued innovations in test and development equipment, continued innovations in detector technology. So today we have an excellent set of partners and supply chain supporters for these technologies, but we're very interested in continuing to see new innovations, particularly in things like micro-optics that will scale at the wafer level, thin profile, high SNR detectors in this wavelength region. You know, there's
we lost again the audio connection, I believe. But I would like to say, first of all, before I give the floor to a couple of questions for you, Benjamin, it means the world to us to have the support of Rockley Photonics in Optica. I know that times had not been easy for Rockley Photonics, but I would like to hear from here, say hello to Aaron, the CTO, and to say hello to Andrew, the CEO. You guys, for many years, have illuminated the silicon photonics landscape, opening the new applications in the biosensing market. And we are more than happy to have you here with us. And thank you so much. Thank you so much, Benjamin. There is a company that I want you to meet, which is Sanguis, Jeffrey. But before I go to him, there are 3 questions that I want to address in the room. The first one is coming all the way from the beautiful city of Lausanne, Ligentech, although I know Anton Vasiliev is actually based in Ghent. Anton, what's on your mind?
Well, thank you, Jose. I'm actually also based in Lausanne right now, so I'm moved to this beautiful city. Thank you, Benjamin, for a nice presentation. Very awesome to see how this device is really being valued to the complex market as well. I'm very curious to know, so your device laser output will heavily depend also on the temperature, I guess, of the actual die in the chip. Do you use active stabilization of the temperature on the wristband, and how precise does it need to be?
Good question. So the precision depends on the application in terms of what level of fidelity that you need. Today, our PIC includes the ability to monitor. So we do at wafer level, we do test and characterization of the wavelengths, so those are known. And then on the PIC, we have the ability to monitor wavelength shifts, as well, of course, the temperature of the silicon itself. So we have good knowledge of the wavelength at the time of measurement. And because the system is quite stable, it's a DBR-based laser. The drift with temperature is relatively small, and we can compensate for it analytically, as long as we know what it is. We also take advantage of the fact that, unlike telecom, I am strapping the sensor to a nice warm mammal. So the temperature range you have to span while it's there is a bit limited.
Thank you very much. From the beautiful city of Lausanne, sorry, Anton, I thought you were still in Ghent. From the beautiful city of Lausanne, we go to an even more beautiful, sorry, Lausanne, Quebec in Canada to meet Castor Optics, Optica Corporate member. Thank you very much for being with us. Tell us, please, Kathy, what's on your mind?
Hi, good morning or good evening or afternoon. Can you hear me?
Yeah, loud and clear.
Yes. Perfect. So my question was, it seemed from your first slide, you're focusing on wristbands for your wearable. Are you considering any other form factor or other types of wearable for your technology?
Yeah, so I covered the bioptics bands specifically because those are public-facing products that we're developing. We do have other form factors in process depending on the needs of the partner. And then we also, in addition to wearables, there are other biosensing applications off the human body that are also of interest using our chips. Did you have a specific one in mind that you wanted to perhaps talk about?
No, I'm just educating myself. Thank you. It's great to see the progress that the technology has come so far in detecting analytes in vivo and non-invasively. So it's great to see the science progress so far.
Thank you.
There is, Benjamin, 2 companies which we thought, the technology scouting team of Optica thought that they could do very well business for you. Those are Sanguis Corporation and Atomica. Let's go first to Sanguis Corporation. Jeffrey, thank you very much for the first time at the Optica Online Industry Meetings. Sanguis Corporation. Jeff, please introduce yourself. You have a couple of questions for Benjamin, and you also brought 1 slide to the meeting.
Okay, I'm going to share the slide. And can everyone see the slide now?
Gorilla Glass clear.
Okay, great. Okay. Basically, yeah, thank you, Helena, Olga, and Elise for the opportunity. Sanguis Corporation is a New York startup that I co-founded several years ago. I'm the chief scientific officer and inventor. We're developing basically advanced Raman technology, Raman spectroscopy, that's perfect for biosensing both in vitro and in vivo, including transdermally. Focus is on sensitive low noise spectrometers and probes that make biosensing and the detection of weakly scattering analytes in turbid media practical. Currently, we're developing a new spectrometer that will have much greater sensitivity on par with like Scheres substrates, but without substrates, and will make Raman pulse plethysmography possible due to its speed. It also lends itself to miniaturization. I was interested in what Rockley was saying because we might need chips made. And for the assessment of blood analytes such as glucose, carotenoid, CRPA1C, and all the rest. It may even be able to detect blood markers for long COVID, all non-invasively, transdermally. We've already tested several dozen patients with our earlier spectrometer and obtained a strong correlation between the spectrometric results and a glucose meter. We also tested glucose solutions down to the hypoglycemic range, and that's what you can see down here. Based on a single simple sum, no curve fitting of the three most significant peaks in the glucose Raman spectrum versus known glucose concentrations. That's what this chart is that you can see. The same novel spectrometer has been tested, by the way, with foods and with samples of hard to detect trace analytes from our Antarctic ice, for example, where there are trace quantities of PFOS, PFAS, DOC, and we could quantify them with a greater than 0.97 correlation with the devices and chemistry that they were using to detect these things. All of our technology has been patented in the US and foreign, and we're confident that our innovations can significantly impact multiple industries. Same goes for the biomedical device based on the technology, that it would enhance the lives of millions, be environmentally sustainable, no medical waste. And right now, we're basically looking to try to commercialize our spectrometers and probes and want to further develop our biomedical device and get that to market. Hence, we're seeking partners, industry partners with an established presence in the marketplace with whom we can work on further development and application, as well as license this technology too. Currently, all the devices and everything are large optical benchtop devices that I could build in the lab here. So we haven't tried to miniaturize anything. But we have got excellent results, as you can see, in the very low hypoglycemic range with glucose, where the measurements are almost spot on. So I'm finished.
Jeffrey, thank you so much for coming to the meeting. Welcome to Optica. This group of people want to help you do business.
Yeah.
Helena told me that you really can do business with Rockley Photonics, and I can see that's obvious. I saw that you had two questions for Benjamin.
Yes. Basically, we're doing Raman, not infrared. And it is in the region that penetrates the skin, no problem with water, like 600 to 900 nanometers.
Yep.
But we need tunable over small ranges, several lasers, like 10 lasers, say, tunable of over maybe 5 nanometers or less for Raman spectroscopy, together with sensors, photodiodes, that would have to be coated with interference coating for filters. Laser blockage.
That's the laser line. No, good. Yep.
And the photodiode, the lasers would need to be RF modulated at different frequencies, actually, low, like 1 megahertz to 5 megahertz range. And the photodiodes would need to have tuned or resonant electronics attached to them to filter out unwanted frequencies.
Mm-hmm.
So we would eventually, down the road, want to make a chip, even if it was a slightly large device that could be a wearable, even a larger wearable, that would basically have maybe 20 lasers of different wavelengths on it, some of them tunable like this, and a bunch of fairly large area photodiodes on the same chip with all the integrated electronics. That could basically emulate what we're doing in a large benchtop system with multiple separate lasers being with fiber combiners and all the rest.
Benjamin, can you help?
Sure. If you send me an email, we can talk about opportunities there. The way that you're talking about is a different, we do have the ability to make photonic integrated circuits in those wavelength regions. It's a different substrate than we're using for our shortwave infrared sensing technology.
Right.
But yeah, happy to talk with you. I actually have some colleagues on staff who, did their postdocs on non-invasive Raman. So we have, we understand what you're working on and some of the challenges. So happy
Excellent. Werner Mantele, all the way from Main. So sorry, from Frankfurt am Main. Please tell us, Werner, what's on your mind? You raised your hand.
Thanks, Jeff, for this presentation. Can we go back to your slide, please?
Yeah, of course. Let me put it back up.
Okay.
Screen, and we'll get it on. Should come on now. There
the
goes.
correlation you show here was made on glucose solutions, correct?
That was made on glucose solutions. Using an apparatus looks somewhat like this, but of course the vial would be clear. This actually is a natto oil from the seeds. So it's measuring carotenoids in the illustration.
Now, if you go for real skin experiments, what is scattering? What is the correlation then between the real concentration and the measured concentration from Raman spectra?
We're getting right now with very crude instrumentation. This is the old spectrometer. There's several spectrometers. The new one is about anywhere from 100 to several thousand times more sensitive, using stimulated Raman, among other things. Very interesting variation. This is with the old spectrometer and the vials in the low physiological range, like 50, milligrams per deciliter.
Okay. And then
the skin, the turbidity of the skin and all of this problem doesn't really affect it, but it adds a lot of unwanted signals. However,
yes, I guess so. Pre-resonance signals from other components of skin. Probably.
Right. There's a lot of very strong signals that weaken, that produce more noise on this curve. You can still get a correlation. It's about 0.85 to 0.9, which isn't bad, without having specific calibration. And but what the new technology, the new spectrometer I'm building, this one uses a CCD with dispersion, although the fixed pattern noise is 100-fold lower than commercial products out there right now. So you don't have the gain variations over wavelength. But
thank you very much, Jeffrey, and we'll come back to you. But there's somebody that I want Benjamin to meet.
The new spectrometer pulse plethysmography avoids the issue of the skin in Raman.
I'll come back to you about that later. I have somebody else who wants to talk to you about that, because I want Benjamin to meet somebody else. I want to go now. We went from New York and we go all the way to Santa Barbara. Atomica for the first time at an Optica Online Industry Meeting. Brian Marr, thank you very much for joining us. The CEO of Atomica. Tell us what Atomica is and ask your question to Benjamin.
Okay, sure. I'll share my slide here.
Atomica is a MEMS manufacturer.
Excellent.
Okay, can you guys see my slide?
Crystal clear.
Okay. Yeah, I'm Brian Merritz. I'm the CTO of Atomica, and we are the largest MEMS fab in the US. And I just wrote our mission statement out here. Our customers are on missions to cure disease and revolutionize sensing and communications, and we are resolved to get them there. We process on both 6- and 8-inch wafers, primarily silicon. We do a lot of glass wafers as well as some other exotic substrates. And anything you'd expect from a MEMS fab, we can do here in Santa Barbara. And I just, so the question was, how can Atomica members accelerate what we do? Well, it's pretty straightforward for a foundry. Come to us with your projects. And so I just wanted to go through 3 kind of examples of things we've done here that may be relevant to biosensing, as these were all biosensing type applications that we were able to adapt. The first example, let's see, sorry, my screen is, oh, there we go, is a laser micro package, which was used as an optical engine. This technology was initially developed for data center communication, telecommunication type applications, but we've recently adapted it to be used in a biosensing application. And basically, it's just a hermetically sealed package where you can put a laser and some other micro optics and use it as a light engine for different applications. We use a proprietary process to seal the wafer, to seal all the lasers in the wafer at the same time. So we get thousands of these chips on a wafer, so we can get good economies of scale. And we hermetically package these at the wafer level, which also allows us to do wafer level testing. So we can get yield out at the wafer level and do that test quickly. And one thing I wanted to mention about this one is, we've been working on this technology for a while. We've shipped millions of these parts, and recently we've also been improving the process. And recently we've had a lot of wafers that came out with 100% yield across all wafers. So we're really proud of the improvements we've made there, and it's a technology we've been doing for years, and it's a pretty mature technology. So if you need an optical engine that's hermetically sealed, this is a good solution for that. The next one I want to talk about is just an example where we have microfluidics, we have MEMS, and we have optics all integrated into one device. This is a cell sorter with an optically addressable channel. So we can look at cells as they travel through the channel and decide based on fluorescent markers whether to collect or allow those cells to pass to the waste stream. And this is a technology that was developed here, and it's now a product for our customer. So we manufacture these MEMS chips for them with the microfluidic channel, with vias through the wafer to the backside for the waste stream, the collection path, and the input, and with an electromagnetically actuated valve. And one thing I wanted to mention on this one is over the last few years, we've really improved the process here. We've recently qualified a new process that we worked with the customer to develop that reduced one whole wafer from the stack. It used to use 4 different wafers, and now we only use 3. And we have a glass wafer on top, so there's an optically adjustable channel. And we reduced the process steps by about 40%, so improving cycle time and cost. And we're really proud of that recent qualification. And the last kind of area that I want to talk about, where we do work with a lot of different companies that are doing different types of biosensing, is in CMOS, or silicon photonics post-processing. CMOS fabs, I used to work in one, are very restrictive in what they can do. And so we'll do a lot of the things that they can't do, and things that, as a MEMS fab, we've gained expertise in through the different programs that we've done here. So obviously we can do precious metals here. We don't have the same restrictions on precious metals. So adding things like gold, silicon, or gold tin, or platinum to either CMOS or silicon photonics wafers. Backside wafer processing, deep silicon etching, either on the front side or the back side.
We are very impressed. Basically, what you can do is a laser on a package. You can integrate microfluidic MEMS and optics, and you can process them. You can manufacture a wafer level and a semiconductor fab. Very impressive, Brian.
Right.
You have a question for Benjamin.
Yeah, I was just curious. You mentioned in one of your slides. I'll stop sharing here, but you mentioned in one of your slides that you are able to, or in your next generation, you're going to have laser attached, flip chip attached without solder or solderless. And I was just curious what that was referring to.
Gotcha. So I believe my colleague Aaron Zilke has published and given some talks on the stamping technology that we use for that. And so if you have questions on that, you guys can hit me up and I can put you in contact with the right experts in-house to describe.
And I know Aaron Zilke is watching, so I would like to say hello to my very good friend. Brian, welcome to Optica, and Benjamin, you guys are fantastic. And honestly, I have to love every member individually the same, but I love you a little bit more than the average Rockley Photonics. Thank you so much. And hello, Andrew Rickman, you are really fantastic. Benjamin, thank you so much. And we move ahead. We continue. And you know what? We have been to Santa Barbara. We have been to New York already. We have been to many places so far. We go now to a really amazing place. We go to Cyprus. And why do we go to Cyprus? Because we want to meet for the first time at an Optica Online Industry Meeting, the company Biolum. Biolum is a biosensor manufacturer and is represented today by the principal engineer, Thanasis Manolis. Thanasis, thank you so much for bringing Cyprus to the global Optica network that industry, that Optica is. The floor and the attention of everyone goes to Biolum.
Thank you. Thank you for the introduction. Let me share my screen. Yeah, so hello, everyone. Sorry. It's actually an honor to be here today. In the next few slides, I'll try to briefly describe our technology and activities at Biolum throughout the years. So Biolum is a young deep tech startup located in Nicosia, Cyprus, operating since 2020. I could not think of a better way to start my presentation than a brief introduction to our team. More specifically, Biolum currently consists of 5 people, as you can see here. Our CEO, Dimitris, our principal scientist, George, Mahmoud, who's our photonics engineer, and Christian, our youngest member, who's our bioengineer. So we're covering almost the full spectrum of our current activities in-house, from mask design to surface functionalization to test and measurement. Now, our mission is to develop the necessary diagnostic tools for early diagnosis and treatment of acute infections at the point of need, and more specifically, sepsis. Sepsis is one of the most significant health complications from pathogen infections that can lead to tissue damage, organ failure, amputations, and eventually death. Sepsis affects more than 48 million people each year and takes 11 million lives worldwide, costing billions of euros annually. So what we should do, what should the clinicians actually do for sepsis patient survival? The answer is to start appropriate and effective antimicrobial treatment within the first hours of the infection, which classifies sepsis as a highly time-dependent disease. Now, our solution targets to address exactly that. Here is a conceptual prototype of how we envision our disposable chip and reader instrument will look like in the future. Our goal is to deliver affordable yet powerful biochips based on proprietary silicon plasmaphotonic technology embedded in a disposable cartridge in order to scan blood for multiple pathogens and biomarkers within minutes. So we want actually to offer a powerful tool to the clinicians to identify blood infections at an early stage in order to start the appropriate treatment to the patient. Now, going deeper to our technology, this is our biosensor concept. It's a silicon nitride Mach-Zehnder interferometer that integrates an aluminum plasmonic transducer in the sensing arm while having some active elements on the reference arm for sensor calibration. The actual transducer is a 7 by 70 micrometer long aluminum plasmonic stripe that enables light propagation on top. Here on the bottom right corner, you can see an SEM image of the actual fabricated device. And I would like to close my presentation with some highlights of our technology so far. So basically, we're using CMOS compatible materials like aluminum for our plasmonic transducer and silicon nitride for the rest of the circuitry. So we can fabricate our chips with optical lithography in wafers. Its biochip is a 20 by 20 millimeter silicon chip that can host multiple copies of our Mach-Zehnder biosensor we biofunctionalize our aluminum surface using in-house chemistries. So antibodies can be immobilized on top, targeting for pathogen and protein detection on the same chip. Now, some of our recent achievements include E. coli detection down to 10 cells per mil by capturing the whole bacterium, and CRP detection in serum samples in the range of 1 to 40 micrograms. So overall, I would say Biolum technology so far can offer a low-cost disposable chip using the CMOS technologies, fast detection, time to result is within minutes, and it's easily scalable, which can be attributed to the ultra small footprint of the aluminum plasmonic transducer. Of course, multiplexing is also an attribute of our chip, so we can actually capture multiple targets like pathogens or proteins on the same chip. So with that, finish my presentation, and I'm open to questions and comments.
Thank you so much. That was a very good presentation, and this is something that is quite important, because effectively, I mean, sepsis is a very big problem, and the amount of that is unbelievable. And one of the things is in the early diagnostics. So do we have here Legentech Anton? So can you say something about how it's going to be these silicon nitride peaks?
Yeah, this is really amazing that this fits so well with what we do, what we are trying to offer to customers and startups such as you are. And it's really nice to see that your technology is really pushing it further to solve a real world problem. So maybe I'll just share one slide. Well, for me, it was a bit difficult to put everything on slides. So please, I'll share it a few slides with the same time. So we are.
There is some background noise. Okay, I think.
Okay. So here at Legentech, we are originally a Swiss company. We are already expanding our presence a little bit over across Europe. So we have an office also close to Paris, where we're in close proximity to X-FAB. And together with our partnership with X-FAB, we are actually right now the largest silicon nitride open access foundry in the European Union. So that, I think, if you want to scale your technology, we are at the right place to enable you to produce millions. Well, I hope we'll not have millions of sepsis cases, but we want to produce. We can produce a lot of devices for you if you're interested. So we have a office here in Switzerland, but I'll just show one application that we have people working with. It's similar to what you are doing, where we expose the waveguides of silicon nitrides locally, such that we can functionalize them. So no, we do not functionalize them. So the other partners, the application partners, such as yourself, functionalize those chips with their proprietary technology that allows them to use them for sensing applications. It's for lab on a chip, like you are doing, not only for bio applications, but also for electronic nose applications. So we are also part of Photonics Fab. So we are the silicon nitride foundry within this consortium, which aims to organize a whole supply chain of devices, next generation devices in Europe. So Aribal is one of our partners who's also working on silicon nitride, and they're doing a very interesting device. It uses silicon nitride where they functionalize the chip in such a way with different sections, such that you can mimic an electronic nose. So if you have a smell, you can start doing PCA analysis of that data to figure out what is exactly now the smell that you're smelling without actually smelling it. So which is very, very interesting. So a bit more about ourselves, and here I'll conclude. So we can support you or others in a seamless journey from idea prototype to high volume. So we have a low barrier entry model where you can participate in the PW runs, where you run on certain stacks that are fixed, they're reliable, that are measured, and you have BDK from it. On top of that, if you want to have something special, like you mentioned, you need aluminum nitride, aluminum for your chips, we can develop a custom process together with you, that would allow us to bring this into a 200mm fab that would allow you to produce many chips at a very efficient price point.
Thank you so much, Anton. This is actually very good. I mean, it's like all the integration is key in all the biosensing and all the technology that we are discussing today. We also have in the room, we also have Sebastian Hook from Tech 5, and I know he's doing also virus detectors like COVID and other kind of virus. Sebastian, are you around?
Yes, I am. Can you hear me?
Yeah. So could you please share your slide and take us a little bit on this diagnosis thing? Thank you.
All right. Can you see my screen?
Now I can see it. Yeah, very clear.
Okay, perfect. So yeah. So
tell us a little bit about you. Yeah.
Yeah. So my name is Sebastian Hülk. I'm the CEO of a company called Tech5 USA. So I'm usually located on beautiful Long Island, New York, to say in the same way like Jose might introduce it, but I'm currently dialing in from Frankfurt because this is where our headquarters is. So we design and produce UV-Vis, NIR, and Raman process spectrometers, but we can also use them in the lab, as you can see here. So in conjunction with UGA, the University of Georgia, we've developed a SERS sensor, so surface enhanced Raman spectroscopy, for SARS-CoV-2 virus detection. You can also use it for different viruses. So in this case, it's an indirect version. So it's an ACE2 capture coupling to the RBD domain of the virus, but it also works as RNA, and in the meantime, also as direct virus detection system. So it's very rapid. So currently we are at like 5 minutes. If you fully automate it, which you're intending to do, you can bring it down to probably 1 minute. It's highly specific. What does it mean? We're currently at 99.8%, but I think we can probably make it even better. And what's really unique about it, it's measuring both qualitatively as well as quantitatively. What do we need for it? So it's a Raman spectrometer that we've designed, also the optical interface and the SERS substrate. We also have mass production capabilities with OAD, which is oblique angle deposition methodologies. And then we've developed actually UGA in this case, the machine learning algorithms to interpret the signals. And as I said before, you can fully automate it. So if you wanted to have a high throughput immunoassay, we could do that. And on the right-hand side, I'm just showing the different steps. So at A, you can see the SERS substrate. So these slanted nanorods made of silver in this case. We also need to have some argon ion etching just to remove contaminants. And then we apply the specific antibody to the nanorods. So in this case, the ACE2, like you would find in a human cell. And then add a nasal swab. You can also use saliva, whatever you want, into, like shown here, a PCR tube. And then we incubate the system. So we bring the SERS substrate into this PCR tube. And if the coronavirus or any other virus, we've tested that with influenza and also more benign NS63 and various others. And if that virus is present, it would bind to the ACE2 cell. And then we wash and dry the substrate, place the whole thing into a Raman spectrometer, use the XYZ table we've developed for that, and also the specific optical interface for it so that we can position the laser. By the way, we use a 785 nanometer Raman laser for this application, and then take the spectra, interpret it, and can say if it's positive or negative, and in addition, what the virus load is, which is quite important, as you know, when it comes to determining how sick that patient might really get. So it's just one slide here. It's obviously way more if you're interested in the technology and how we could fully automate it. I just wanted to limit it to a brief presentation here.
Yeah, thank you. Thank you so much. Yeah, go ahead. Sorry.
Yeah, no, thank you very much. I would like to acknowledge that Tech5 is a corporate member of Optica, and the way that they are here presenting this, Tech5 is an spectroscopy company, and they're looking for partners. They're looking for partners to further develop this and take this to the market. Could you tell us a bit about what kind of partners are you looking for? How we can help you, Sebastian?
Yeah, or actually, if you want to produce with us, so we got 4 business units. We got all the photonics components and building blocks from various benches. I think our core expertise is the electronics that we've developed. So we have quite some business in the semiconductor business. If you think about multi-layer thin film thickness analysis by means of spectroscopic ellipsometry and interferometry, this is where we're really big at. So we've developed the electronics to read out the analog signals of the imaging sensor in high speeds, digitize it, and then also all the interface protocols from Modbus to Profibus to OPC UA, you name it, depending on what they use in the factory. So we're really in semiconductor manufacturing equipment, in food, feed, and beverages, in oil and gas, in chemistry, and in pharma and biotech, as the sample or the example here shows today. And then business unit number 2, we're designing fully fledged turnkey solutions, so online analyzers that go into factories, to refineries, to chemical factories, etc. And then number 3, we got some distribution going on. So we belong to the so-called Dynamic Photonics Group. It's a company existing of 11 in total, traded at the Frankfurt Stock Exchange. You might know a company called Spectral Engines. They have developed also Fabry, Perot interferometers in MEMS technology. We use them for quite a successful product called TactiScan for illicit drug detection. So that's what we're rolling out all the stage right now, from heroin to cocaine to fentanyl detection, all the different mixtures, including the cutting agent, so we can detect that. And then
thank you very much, Sebastian. If you want to help Tech5, if you can help Tech5, please let us know. They are looking for partners, and they are here representing the Mnemonic Group. And I also would like to say hello to Ben Oderkerk, former CEO of Avantes, who is the one who introduced me to this fantastic group. Thank you, Sebastian. Let me continue. And before we give the floor to Laura Lechuga, I know you all are waiting to hear her. I want to go to Aston University. I want to go to United Kingdom. I want to go to Aston University, because now is when we are going to start talking about glucose sensing. And I think all of you know Professor Edik Rafailov. If you don't know him, I would like to introduce you to a very dear friend of mine. Edik, thank you for joining us all the way from Aston University. Tell us, how do you measure blood and how do you measure glucose in wearable devices? Edik Rafailov, we cannot hear you. You're on mute. That's the sentence of the pandemic. You are on mute. Do you remember the pandemic? Oh my God, they're saying COVID is back. Edik, we can hear you now.
Can you hear me?
Crystal clear.
No, unfortunately, it's not working.
I will come back to you after Laura Lechuga. Edik, I'm coming back to you after Laura Lechuga. And now, the moment that you've been waiting for, our keynote speaker today, the person that anybody in biosensing has come across and has been inspired by, I would like to give the floor to the head of the nano biosensors and bioanalytical applications of the ICN2, all the way from Spain, my dear country, my dear Laura Lechuga. The floor is yours.
Hello to everybody. Can you hear me? Hola, Jose.
Very clearly. Hola.
Okay, so well, thank you for the invitation. Thank you for giving me the opportunity to tell about our work on nanophotonic biosensors. I have been working, I think, on nanophotonic biosensor all my life, almost 20 years. So today I am going to focus how we develop this nanophotonic biosensor, mainly for the diagnostics of bacterial infection. I think already one of my colleagues has already introduced you the problem that we are facing. We call this the silence pandemic. And we know that sepsis, not only sepsis, but also the antimicrobial resistant infections due to resistant bacteria is a major concern. We know that in the case of sepsis, for example, in 2017, we have more than 11 million of deaths and death, more than 40 million of cases, but this mortality is increasing every year. So this is you can see here how what is my colleagues in the hospital, they are just finding today this one that someone is coming with an infection to the hospital, and they see that most of the, they know that they have infection by a bacteria, but then the bacteria is completely resistant to any of the antibiotics that we have in the world. So the situation is such a way that we know that by the year 2000, the predictions say that by the year 2050, probably the number of deaths due to this antimicrobial resistant infection due to sepsis is going to be even more than for cancer. So in 2050, every 3 seconds, a person will die due to an infection. So as one of my colleagues say before, main problem that we have in our hospital, we have excellent techniques, but you know what happens is all these techniques are not enough rapid. Since a person arrives to the hospital, there is an antibiotic code, and then there is a suspected infection. Once they take the sample, it takes between several days, even a complete 1 week, to know not only what is the pathogen infecting the person, but also what is the antimicrobial resistant profile. So to know which one is the right antibiotic that we have to deliver to the patient. So we want to change this, and we want to save time. And then we want to provide a technology where we can move from days to less than 1 hour in order to, it's the only way to be able to give the right treatment to the patient. So how we are doing that? So we are doing that in this way. So we are just using our patented silicon photonics technology. Probably if you know my work, we have been working with a new concept that we introduced several years ago, the bimodal waveguide interferometric biosensor. This is a biosensor and interferometric one just fabricating in silicon technology. We have a very high sensitivity, provided real-time, level-free, is working in the visible range with a multiplex configuration, and we can afford a limit of detection in the picomolar, femtomolar in a label-free way without any amplification. So this is what we're working just now. So we want just to develop on this multiplex biosensor chip, and we want to provide 3 different biochips. So the first one is going to detect in a very sensitive way which bacteria is infecting the patient. We are going to have a second chip where we can do a very rapid antibiotic susceptibility test, so we can know which is the best the suitable antibiotic for the person for this infection. And we are fabricating also a third biosensor where we make a monitoring of the antimicrobial therapy. So what is called the therapeutic drug monitoring. And we account with many with many results already. So my group has been demonstrated during the several years ago that we are able just to make a very fast bacteria identification and quantification, one single step, just directly in the real sample from patients. And we can have, depending on the bacteria, and depending on the quality of the antibodies that we're using, the selectivity, we can go even for limit of detection below 4 CFU per milliliter. So mean that our technology has the capability to detect even one single bacteria. Results are always done in acidic, for example, this one, acidic fluid from patients. You can read also our work, how we detect sepsis and how we develop point-of-care devices going to the hospital and detected the sepsis in less than 10 minutes, just on-site in the hospital. But we have already also many results in a second biosensor, how we can detect, one, we know the bacteria, what is the pathogen infecting. We can detect also what is the profile of the resistant bacteria profile. So we can detect which bacteria, for example, this is a result between Staphylococcus aureus, the resistant and the non-resistant one. We can also develop a biosensor where we can detect in a very fast way what is the antimicrobial resistant genes. This is also from E. coli. You can read also in our publication with an astonished limit of detection of atomolar level. And also we have recently also developed another biosensor we can afford this therapeutic drug monitoring. So we know the pathogen, we know the resistant profile of this pathogen, and then we know which is the antibiotic that we have to supply to the patient. But what we want to do is also to have a continuous monitoring. How is the concentration of this antibiotic in the blood of the patient in order to adjust the dose that we need for the patient in order to recover as soon as possible. So what the time to result to each of these biosensor is less than 30 minutes each of them. So our next, so we are trying to move, what we like is to moving our biosensor technology real to market. So we have mainly laboratory setups and devices. We are able to fabricate all the sensor chips in even a multiplex configuration, microfluidics. We are really expert in the surface biofunctionalization. We have a new technique for the biochip packaging. I mean, once we have the sensor and with the all the bioreceptor on top, we have developed all the optical system, electronics, software, and so on. But we need to move this to a commercial product. And we are just starting just now a new company called Heroica Diagnostics. And we'd like to make this this biosensor a reality. So the idea is that we can provide one device with many different cartridges, with many different application biochips, depending on the pathology. And in any case, we want to contribute to develop this personalized medicine. And our promise is that we can go from days to less than 1 hour, especially in the case of the infections. And of course, my group has been working for many years. So here is a summary of all the different bio applications that we have been able to develop. So just in case you are interested in any of them, because we are working not only in health, but also in environmental applications. And this is just a summary of our capabilities, mainly in the application domain, but also we are expert in the fabrication of the photonic biosensors. So thank you.
Thank you so much, Laura. It's really, really impressive because the sensitivity that you are giving and the time response that you are giving is quite fast. It's actually very competitive with mass spectrometry, which is usually the traditional technique. So this is so much. I mean, like if you say one bacteria, and that's a big deal. It's a big change. I would think it's going to be a big leap in this. So you obviously are looking for partnership, but now. So we have here Eric Rafaelov, which was before. So Eric, are you here? So what do you think about this? Because I mean, you have a lot of insights to put in there. Eric, are you there? I think
Yes.
Eric.
Hello? Can you
Okay.
hear me?
Yes. Yeah.
Yes, but I would like to, just because this webinar, it's a biosensor, biological sensor development. And I would like to present what we developed, and quite possible maybe you will be interested in that, because we have demonstrated useful kind of application of that. And if you give me a floor, I will do that. Because in our case, we not measure, because what we're trying to do, we're trying to develop a real non-invasive device. In our case, we focus on a laser kind of base devices, not LED, because one of the effect what we're exploiting, it is a laser Doppler fluorometry, which give us information about blood perfusion. And at the same time, we now start to develop multi-channel system. And in our case, we introduce autofluorescence technique, which give us a metabolic activity. And in this case, what we did that we already demonstrated that these devices can be extremely useful for monitoring diseases like diabetes. And this one, it can be helpful for clinician to monitor medication of the patient. Because these devices can be transferred remotely, send information to the GP or doctors. And this one, it's why in our case, we're just focusing on a non-invasive without any blood testing or something like that. Of course, it's this technology, what we're proposing, Jose maybe slightly been a little bit optimistic to say that we can monitor glucose. No, sorry, Jose, we cannot monitor glucose yet. but at the same time, technology what we develop is a good background to develop non-invasive blood pressure measurement with high precision. And this one what we're working on at the moment. And yeah, I have a few slides just to show how looks our device. If you give
Yeah,
me
Eric, you have a lot of slides, I know, and I have seen your presentations. They're fantastic. Can you show us one slide that summarizes the concept, and then we can go to Laura to see how you can work together?
No, I don't want to do it. Just okay, share. Where's share? Just a moment, I will try.
By the way, many of you know Eric Rafailov, and if you don't, and you meet him at the conference, have a chat with him. This is one of the most exciting people I know. Eric, tell us.
Can you see that? I don't know.
Yes, go to presentation mode, and then we'll see it a bit bigger.
Oh, okay. Okay, yeah, this one is
and
a
you're
main idea.
the wrong screen, so you go to display settings, then you can revert.
Okay, let's go again.
This is the way of doing the meetings live. It's when we all manage to.
Yes, I know that. I didn't expect I will perform anything. Can you see
Now
that
we
now?
go perfectly.
Now, this one again, it's a main idea. It's a laser light to just illuminate, and we can pick up it's a blood perfusion, how blood is moving. And we know this a frequency of the all biorhythms that the human body has. And based on that, we can identify intensity of these biorhythms. And based on that, this one is an example of the stuff. And how it looks device, this device, it looks like that. And you can see that compared to the big desktop device, it's just wearable device which you can wear on your wrist. This one, it's the main idea of our technology, and what I'm trying to say that this what we already were developing these devices, and our next step, we're just increasing number of channels, what we can increase functionality of these devices. But at the same time, of course, we would be happy to collaborate with people who are interested in this direction. Yeah, I'm trying to stop because Jose just saying need to save time.
Yeah, thank you very much. No. We have to use the time smartly, but never need to save time. We have time. But Eric, thank you very much for being with us. I want to go back to Laura Lechuga. Laura, we know each other for 20 years now. That means that we are really young, you and me. And I would like to ask you, here you have the entire supply chain of photonics and MEMS. What can they do for you, and what can you do for them?
Okay, so well, probably my main, I have two issues to solve. First is the fabrication, because we are fabricating all our silicon nitride chips with a standard microelectronics fabrication in our clean room in Barcelona. So we have also facilities here, but it's more, it's a production clean room, but in any case, it's for commercial purpose, it could be much more difficult. So this is one. And the second is the integration, the full integration in the point of care device. So the full engineering, full optical engineering and electronics engineering. So this is probably what we're looking for. Or we are more interested in collaborating. Because, we are expert in the biopersonalization. We are expert even in fabricating on the cartridge, microfluidics, so it's something that we can do by ourselves, but we are more interested in the full integration in a complete point of care platform.
All right. I want to also come back to you, Laura, because I know you said that you manufacture your own silicon nitride chips. Could you tell us about the platform that you use at ICN2 and how is the silicon nitride platform? Because we also have Stefan Heinemann here from Lionics, and we have also Legentag, as they presented before. Could you tell us about your silicon nitride platform?
Yes, well, the silicon nitride platform is not from ICN2, it's from the CNM, Microelectronics National Center, and for the Spanish National Research Council, so it's close by to my institute, and it's the large facility that we have in Spain for the fabrication of the silicon nitride chips. So this is very nice for us because we are very close by, and also because we are internal users, so the prices are very low. But okay, this is also, any of you want to develop these silicon nitride chips, we have also this silicon nitride platform that you can see in the website. Link it also to the company BLC in Valencia. So it's the company who is handling all the silicon nitride platform. But as I say, what we're fabricating now, I mean, if we want to go for large scale fabrication, so probably we will need to look for another platform outside Spain.
Thank you very much for being here. And there is somebody I want you to meet. All of you know that this meeting is very important for us because it is a biosensing meeting. We have our technology scouting team actively looking for new success stories to tell us. And we came up with a company. We found a company called Oxiprem. Alexander Nitsch, you are here with us. Please unmute your microphone, switch on your camera. We are really excited to give you the floor right now. This is the equator of the meeting. We still have Surfix, Modulite, and Diamond Tech to tell us how they are helping the world. But now let's pay attention to this. We have a company developing oxygen saturation sensors for monitoring the brain of babies. We are really excited about this. The floor and the attention of everyone goes to Oxiprem.
Of course, we are here, Jose. Thank you for the floor. And I mean, I must say, we're all sitting through lots and lots and lots of these conference calls, you know, with lots of people on the line. And I must say, I'm not sure if I've ever been in a meeting that was as energetic as this one. So, Jose, I was talking to Stefan before, and we said, if it doesn't work in optics for you, you can be great on TV as a show host.
No, my mother says I have a face for the radio, but that's okay. Alexander, please tell us.
Okay, great. Let me share the screen. So this one, you should see my slide. Are you?
Yes, we do.
Yes. Okay, great. So hello, everybody. My name is Alex. I'm a CEO of Oxiprem based in Switzerland. And what we do is basically can be summarized under newborn brain protection. So we use a near-spaced system to essentially perform tissue oximetry. And our first group of patients are prematurely born kids. Now, why have we chosen this group of patients? Fairly simple explanation. Many years ago, that was before 2010, the Biomedical Optics Research Lab here in Zurich under Martin Wolf got approached by a large group of neonatal and pediatric specialists, and they say, hey, we can monitor virtually everything in these kids, but the one thing that we don't see with the precision and the accuracy that we need is the brain oxygen levels. So the Biomedical Optics Lab took up the challenge many years ago. Also, our CTO, Stefan Kleiser, is on the call, so he can also answer questions later on. And yeah, basically what makes us different and what made our mission so special was that we had to design a reusable sensor that is special in a way that most sensors that you find otherwise are disposable. Disposable sensors are cool solutions, but having a disposable device always puts restrictions on you in terms of costs, in terms of hardware. How elaborate can it be that you're making? So for us, it was very clear that we had to go down the reusable road if we wanted the precision that we were asked for, basically. And the result from that is that our sensor that we are preparing to launch into the market in sometime between 1 and 1.5 years from now has 13 wavelengths to interrogate the tissue. So that is versus 2 to 5 wavelengths that disposable systems can do. And also on the algorithm side, we have come up with an algorithm that is self-calibrating, that makes all the readings much more robust against things like skin inhomogeneities, hair, and all this kind of stuff. And on the picture that you see on top, actually you see a baby here in the University Hospital in Zurich at just 970 grams, and you see our sensor on the head of that child, and you see the hand of the nurse next to that, so the size impression is quite obvious. So we are packing a lot of technology into a very small space. Yeah, well, I think for my 2 minutes, that should be fine. Since we all love working prototypes. So on the bottom left corner, this QR code is our recent prototype. If you scan that, it should actually create an email that you can just fire away. Then we've got your contact details and can provide you with more information on Oxiprem, on our journey, on our technology. And you're all very much invited to do
If
that.
you miss the QR code, all you have to do is send us an email and we make the introduction for you. Alexander, welcome to Optica. Thank you for joining as a corporate member. It is great to have you in the room. Alexander, can you remind us which wavelength range do you use?
Yes, so we are from the mid 500s to 1000 and Stefan, correct me, 1060? Yeah.
Mid 500 to 1060. Just pay attention to this. These guys actually put arrays of LEDs and arrays of detectors, and they managed to synchronize them. So this is a really amazing technology, and they have actually been certified already in their first pilot run. So these guys already have hospitals demanding and actually hospitals using these oxygen sensors. Alexander, how can we help you? You have everyone here, the lasers, the detectors, the LEDs, the packaging, the hospitals. Tell us, how can we help you?
Well, that I will gladly pass over to Stefan because he's the CTO and
is he there?
Of course he is here.
I love this. This is how startups work. You know, is Stefan here and then he unlocks the camera. Stefan,
actually,
tell us,
he's just 4 meters behind me, so.
All right.
Stefan?
Yeah, generally, I think, we can always reach out to other suppliers. As you can imagine, with so many wavelengths, we rely on custom LEDs that have the wavelength we need, which are not so super standard, of course. Regarding detectors, we're also looking at highly sensitive ones, still ones that can be fairly easily soldered onto PCBs. And in terms of encasing, we're right now in the domain of casting silicone around the flexible electronics. So I mean, generally, we're always out looking for additional suppliers and always trying to improve on the technology side. So if you supply any of that, be happy to have your contacts. Yeah.
Thank you very much, Stefan. You know Oxiprem joined Optica because there's something that you guys know that we do very well. They are looking for investment and we're helping them also with that part. Thank you very much, Oxiprem. There is a question for you coming all the way from Diamond Tech. Werner, tell us, what's on your mind? You are on mute. You're on mute.
Okay,
yes.
Can you hear me now? Yes. I cannot supply you with light sources or detectors, but Alexander, I wanted to ask you, wouldn't it be a possibility to extend the range of wavelengths by like 3 to 4 wavelengths in the blue or near UV to get an access to an important parameter, namely the bilipigments that need to be measured for newborn babies because of the jaundice problem? And that sounds like you could make a double sensor out of that.
It's generally possible to do so. We wouldn't even have to change much in the device. It's just that, all the neonatology departments have already devices measuring bilirubin in the babies because it's a common problem, as you say.
But it's normally invasively measured.
That's true, but they anyway do several they take blood several times a day typically to measure all the other clinical parameters. But I believe what you suggest could be a valuable addition in the future once we have established with our device doing near-infrared spectroscopy. I mean, we literally only would have to add another LED in the blue.
Okay.
Thank you very much. And let me continue with the program today. Started showing you this slide. These are all the companies who registered for this meeting. And then we had the photonic integrated circuit part. If you ask all these people, what is the biggest challenge of making a biosensor with photonic integrated circuits, all of them will tell you it is not the integrated photonics technology, that's mature. It is the chemistry. It is the surface activation. And to do that, we want to introduce you to the company that has worked really hard over the last decade to solve that particular challenge, surface activation of any semiconductor technology, including iterative photonics. I would like to go to the Netherlands, a beautiful country with windmills and with caramel, to introduce Luc Scheres, Surfix Diagnostics. The floor and attention of everyone goes to you.
So thank you, Jose. Also, thanks for introducing our nice country. Yeah, so I'm Luc Scheres. I started the company in 2011. So the first slide is about the company where we are right now. We are located in Wageningen. We're a spin-off from Wageningen University, started in 2011. And indeed, as Jose just explained, we started as a coating chemist, a coating surface chemistry coating company, and we have been doing this for roughly 10 years. In between, we also got we still once got acquired for this coating technology, but during the pandemic, we made a pivot from coating company into a diagnostic company. So in 2021. We got investment to develop this photonic platform. So we started with a team of surface chemists, and we started developing the photonic diagnostics platform, the biosensor technology, and we did some showcase work. So we did, of course, during the pandemic, we did some COVID-19 antibody detections, but then we moved also into the sepsis for IL-6 detection as a showcase. And recently, we started a large commercial project on bladder cancer detection from urine. So we're not a surface chemist company anymore, Jose. I hope you keep this in mind now. So we really moved to developing the platform. And we're focusing on cancer. So cancer, why cancer? Because next to sepsis and antimicrobial resistance, diabetes, also cancer is really a world problem, I would say. The World Health Organization, if you look at these numbers of new cases, what they predict, and also the number of deaths, it's huge, it's amazing. But if you start doing better diagnostics, then definitely you will improve the patient's life, and also reduce some healthcare costs significantly. How we want to do this is with liquid biopsy, so not taking biopsy from a solid tumor, but working from body liquids like urine or blood, for instance. And the nice thing there is that it's quick, it's easy, it's minimally invasive, and it can be applied during the full patient journey. So going from early cancer screening, diagnosis, to therapy selection, and also treatment monitoring. So the platform we're building, how it looks like, you can see on the right images. So what we're aiming for is an accurate system, so high sensitivity and specificity, multiplexing. So it's a small chip with a lot of sensors on it, so we can detect various biomarkers in one test. It's flexible, so it can be done. It can be used for immunoassays, so protein-based testing, but also molecular assays with DNA or RNA. And it can be done on the same instrument. And of course, it should be cheap and simple to use. So on the pictures, you see a €5 coin, and you see some small chips next to it. So we started with a chip that was 5 by 10 millimeters, and we reduced it, we shrinked it to reduce costs as well, but also to the small one, that's a 3 by 3.5, and that small chip we integrated in the cartridge. And below you see a picture of our readout system. It's the first prototype. We are currently, we have 3 of them now, so we're still validating. That's where we are right now. How it works, it's a small chip, and if you zoom in, then you can see the multiplex design. So you have all these spiral structures on it, and 2 spiral structures together makes 1 sensor, so it's a self-referencing system. And then if you even zoom in further, then you can see that the spiral is actually very long, and that means that you have a lot of interaction length with your sample, and that means that we have a very high sensitivity in a multiplex design on a very small footprint chip. And how it actually works. So it's, I think it's rather similar than what Laura Lechuga and also Biolum presented. So we have a surface, light is going through the waveguide from left to right. And if you have, for instance, in this case, antibodies on the surface, if the specific interaction with an antigen is there, then you get an optical shift, and that optical shift we detect. We did quite some proof points already, as I also explained in the first slide. So we did quite a lot of COVID work and then moved to sepsis with IL-6. And what we show there is that we can be very sensitive, also easily to work with human samples, so serum or plasma. And we also did a lot of benchmarking with the Roches and the Abbotts and so on to see how they were performing with their systems in the same assays. And then we see that we are doing pretty well. Yeah. What we do, we started as a coating company, but now we are definitely a bit broader. So the photonic chip technology, we work together with Bionics there. So that coating technology we put over it, that's our what we bring in. We do a lot of biotechnology, so we work a lot with new biotech principles, that's called like that. But it's also about biotechnology, is also about spotting and printing the right biocapture molecules on the sensor. So then we convert, let's say, a bare photonic chip into a biochip, and that's what we integrate into a cartridge technology. Also there we have a lot of experience because we did a lot of coating work for plastic microfluidics. So we do there a lot in coating the channels, not losing your analyte as well. And that cartridge then goes into the readout instrument, and then also a lot of technology comes in. So you need the optical alignment, for instance. You need data analysis. You need smart algorithms to extract the data. You want to have noise reduction and so on. And of course, also mentioned already by Laura, manufacturing is definitely a thing. What we did there already is we developed let's say a workflow that we can do all the chemistry, so the nano coating, and also the introduction, the printing of the biomolecules. We can do on wafer scale, also the dicing we do on wafer scale, and then we use pick and place to assemble it into a cartridge. By combining all these technologies, we hope to detect less. So this is what we're aiming for. This is our development roadmap. So you can see that with the immunoassays, I would say that looking at the clinically relevant concentrations, we're pretty there already. But also the last half a year, we're pushing really hard on the molecular diagnostics, and we hope to reach also the very low concentrations there, so we can have a market access there as well. And that's my story. So if you want to know questions from my side, Jose, then it's really about
thank you so much, Luke. Okay. Yeah, can you hear me? Yes, thank you so much, Luke. This is really good. So I have a very quick question for you. So you are doing this in fluid. So what is it possible to translate this to non-invasive? I mean, you have urine, but non-invasive, like maybe saliva or tears.
Sorry,
I know it's very complex. Is it possible to translate all this technology to other fluids like saliva or tears?
Yeah, sure. Yeah, we did work in milk, but also in saliva, interstitial fluids could work, or sweat.
Sweat, yeah. Saliva is.
Then it's okay for us. But there are examples like what the Gentech was showing of Alibal. That's more the gas phase vapor detection, of course.
Okay. Yes. Yeah. So after this presentation, Laura, tell us, what do you think about this now?
Well, I mean, we have been, Surfix, and we know each other for a long time. So just to comment, Elena, that for a biosensor, you always need a biofluid. I am convinced that this non-invasive technique are very difficult, and you always need a human biofluid in order to make the analysis. This is first. Second, depends on the application. I mean, I mean, you can monitor in saliva, in tear, in sweat, or in blood, or in urine, but depends on your application. I mean, if you're looking for a cancer, probably you need to just to monitor tissue, or you have to monitor blood. But it's very, I mean, but you cannot find anything in saliva or in sweat. So depends on the application. You have to think what is the best biofluid that you need. And the same, I would like to remember also to all of you what a biosensor is. Biosensor means that you have a bio receptor included in the sensing, in the sensor. If not, you are making a chemical sensor. So this is something also that I would like to address here, because when you are speaking about non-invasive detection of glucose, this is a chemical sensor. It's not a physical sensor. It's not a biosensor. Remember that biosensor is much more complicated because you have to introduce a biological receptor. And as Luke knows very well, this is the most difficult part. So even if you think our photonic chips are amazing, are complicated, and so on, the most difficult part, remember, is always how to introduce the correct biological receptor that we need to make a very sensitive and a very specific analysis in a drop of a human biofluid. Remember that, please.
Okay. I fully agree. I also heard you saying that we have a very difficult job.
Yeah, it's the most difficult in this area. All this bio interface is one of the most difficult. So I would like also to offer, if any of you want to collaborate and so on, I mean, we are also at Surfix, we are also experts, so we can collaborate.
What is the biggest challenge? You both are saying, Luke and Laura, this is very difficult. What is the biggest challenge?
Well, the biggest challenge that you need to we want to monitor in a real sample. Imagine that you want to monitor in blood or in urine or in sweat, and you don't want to treat the sample. So you just want to use a crude sample. And you have many interference because in blood, in serum, whatever, you have so many interference. And we are detecting at the femtomolar, attomolar level. So you really need to produce a very sensitive and anti-fouling coating that you are able to detect only the molecule that you want to detect, the analyte, in a very, very complex sample, and also with a very, very low level. So you have very few molecules. Imagine that you want to detect very few virus or bacteria or whatever, but you have hundreds of interference in the same sample. And you need to be very selective.
So look, how far are we to have this anti-fouling coating stable and suitable to volume production?
Well, the point is that there is not any universal recipe. So this is the bad news for everybody, is there is not an universal recipe. Luke knows quite well as me. So you have to develop on purpose for each bioreceptor and for each application. It's possible to make industrial production, but this is an expertise that is kept in each company or in each group. And this is not, it's not, there is not a universal recipe. It's not like producing photonic chip that you go to the clean room and then you can fabricate always the same.
Look, I understand because you're nodding along constantly that you fully agree with everything that Laura is saying. But on the manufacturing side as a company, how far are we to have, at least for one of the big applications, the stable anti-fouling coating that Laura refers to?
Yeah, so I think that the stable anti-fouling coating is not our biggest challenge. That's really in scaling the whole technology. So the coating technology we scaled already to wafer level. The printing of the antibodies or the DNA we scaled already to wafer level. But it's more like we are still on 4-inch wafers. So we have to increase the size of the wafers. You want to do QC on wafer scale because we do all the steps with the biology included already on a wafer scale. So how do we and we have edge incoupling. So how can we do efficiently QC? Because for the IVDR, for the regulatory, you have to follow your process and you want to know which chip is working properly because you cannot make a test that is not performing well. So that I think that is challenging. Maybe to comment a bit on this specificity and the non-specific absorption and the coatings. Maybe it's good to explain it to you, Jose, how it works. We normally call this romantic surfaces. So imagine you are in the bar, you and your wife, and there are 39 other ladies in the bar. And that means that if you have a romantic surface, then your wife is only binding to you and the rest of the ladies is repelled. So that is how it also works in blood or in other samples. I think that's always a good example, and then you get it.
I think I got it. that was a really interesting example. Let me bring to a table, because we're having a very nice discussion between two of my dear friends, Laura and Luke. Let me bring a third person to this group. Anton from Ligentech, what do you have in mind?
Yeah, so I think your biggest challenge is also what we are also trying to address close to Paris and at X-Fab. So there we have our 200mm line, which we're installing different modules to. And one of the questions we also receive from other partners is that, can you do certain qualification? Can you ensure that the process that the processes are tracked? And so the fab there is such is installed in such a way that is automatically fulfilled, right? So you have all the processes that are very rigid. So once you install the module, once you install the process, it will become very rigid and trackable. So you can have a whole track from start to finish of the wafer to your product. So that's something we should really talk to each other also about. How can we help each other there?
But that sounds like the wafer processing of making the wafer. But it gets more complex if you have the chemistry, the biology, and dicing included as well.
Yeah, exactly. So that's also not only the processing of the chip itself, but also the final steps need to be preferably in the same fab, because otherwise, if you start transferring things around, your certifications will probably not be able to. You will not be able to get the certification properly certified. So that's what we're also actively looking into within Photonics Fab, is to enable such a value chain, supply chain, together with all different partners across Europe, that you would have one-stop shop, let's say, where you can go from bare wafer to products, preferably in the same factory, that you can enable your high volume devices. Because as I understand, in your case, you're one of the main sensing players or applications areas where you really need the large volumes, right?
You know, Anton and Luke, Laura has been working with me for many years reviewing European projects. Laura, how close are we to have a peak-based certified product in biosensing?
In biosensing. Well, I think probably now we are much more close after the pandemic, you know, because during the pandemic, everybody just really noticed how useful was to have this COVID test at home that you can do at home. So I think this has accelerated a lot the transfer of the biosensor devices to the market. So I think now we are in the right moment to try to accelerate and try to go to the market as soon as possible. But remember that the idea is not to have a general technology. The idea is that you have to commercialize applications. What people is looking for are only applications. They don't mind even which is the photonics technology that you have behind. They just want a small point of care where you can deploy your drop of sample, press the button, and in a few minutes to have the answer. Okay, so so this is what we want to have in the future.
And we can only do that by working together. You want to get in touch with either Anton, Luke, or Laura, or any other of the speakers, all you have to do is send me an email, jpozo@optica.org, and I will make the introduction. You know what, Laura? Last Sunday was Cancer Research Day. Because of that, one of our corporate members, all the way in Tampere, Finland, beautiful city, swimming the lake if you can, beautiful city, they told us we have made a very big push over the last decade to have technologies for diagnosing and treating cancer. It was very important for us to have at our biosensing meeting the company Modulite. Tommi Hakulinen, thank you very much for being with us today. The floor and the attention of everyone goes to one of the most beautiful cities in this continent, goes to Tampere, Finland. The floor is yours.
Thank you very much for the introduction, Jose. So I'm covering up for our CDO, Petteri Uusimaa, tonight, and pleasure to meet you all, and glad to talk to you a bit what we do. So I will start sharing my screen. So I hope you can see my screen.
Yes, we can.
Perfect. So first of all, shortly about Modulite. So we are in the mission of killing the cancer with science and technology. We are a biomedical company. We are based in Tampere, Finland, indeed, like Jose introduced, with sales process also in the United States. In our customer portfolio, we have a lot of big pharmas and other Fortune 500 companies, and also well-known cancer centers around the globe. We have been around since year 2000, and in 2021 we were listed in Nasdaq First North list in Helsinki Stock Exchange. So we have over 20 years of experience in various laser technologies, and probably where we are most and best known is our work in life sciences, and the key strategic areas are oncology, ophthalmology, genetics, and diagnostics. And we are the only vertically integrated medical laser manufacturer in the world, so that makes us special. So we do lasers from semiconductor chip level up to clinical systems. I would like to showcase the light engine product of ours. So we call this ML6600. So it's a platform product that can host different laser technologies, and it can cover a wide spectrum of wavelengths from UV up to 2 micron. We focus on understanding the customer's application, and then we tailor the product exactly to the customer's specification. What is special on this platform is the cloud connection, and it enables a couple of exciting things like preventive maintenance. So we have a lot of sensors built in the device, so we can monitor the laser parameters as well as also temperature and humidity in the environment, and then get do some predictive analytics and send the customer a notification that please schedule a service call with us should the power, for example, be great. The cloud connection enables also new business models like pay-per-use type of usage. So I think this is also quite unique for our offering. I will show a couple of cases related to biosensing. So first of all, the fluorescence cystoscopy study that we did. So on this slide and also behind me, you can see our newly built biomedical laboratories. They play a vital role in catalyzing the customer R&D, and the laboratories enhance testing of cancer models and light delivery instruments. And this will speed up the customer's journey to the market. In this case, we have tailored the ML7710 clinical laser platform that you can see here on the picture right here. We tailored it for cystoscopy fluorescence imaging, and it was integrated to a photodynamic diagnosis system from Olympus, shown here in the picture on the left-hand side. We conducted a series of experiments optimizing the laser light and the light delivery optics with anatomical bladder model in the biolab. And the laser platform was tailored to include red, green, and blue lasers, which the user can mix to get the perfect illumination. After testing with the anatomical model here, the system was tested with real patients by urologists, shown here on the photo and on the right-hand side. Same laser can be used also for photodynamic therapy with any other drugs the customer has, and also for localizing the tumor using the fluorescence diagnostics. Other showcase that I have is the light source light engine for the flow cytometry application. And in this application, many times the challenge is to build a laser that is stable enough to hit the flow cell repeatedly with the precision of a few microns. And we built a solution where we integrate the laser sources, beam shaping optics, and active beam alignment system to one housing, and this can be used as a light source for flow cytometry. The output of this laser is a readily configured beam ladder, and this can be automatically adjusted over the cloud to hit the target repeatedly and over again. So these showcases were the ones that I wanted to introduce you today, and thank you for your attention, and looking forward to any questions that you might have.
Thank you, Tommy. Thank you so much. So one of the things that we have here is like we have presented in this meeting, particularly techniques based on fluorescence and Raman. So what would you say about that?
Yeah, I think all applications definitely need lasers. So I think they are also very interesting for us. So I would be happy to hear what kind of headaches you might have on the light sources currently. So maybe we would be able to help you out on that.
What the industry can do for you? To give me your biggest challenge.
Biggest challenge. Well, I would be happy to get really a cool application to help the industry. Also, big volume, difficult to solve, and that requires many wavelengths and customized optic solutions. So,
sorry. Yeah.
So he's looking for an application that needs a lot of wavelengths, multi-wavelengths, and we already had a few presentations from Biolum. I want to go to Biolum now. Thanasis Manolis. I think here we have a company who says vertically integrated, they make laser diodes. What is, in your opinion, the difficult wavelength or difficult wavelength range that we can actually target or we can challenge Modulite?
Well, our technology, we're currently working at telecom wavelengths 1550. It's quite challenging also to do this maybe on chip. However, we are open to, it's actually an ongoing work currently on Biolum to design maybe an optical readout for our sensor. We also have external collaborators, but we are also open to discussions specifically maybe for parts like high resolution, maybe infrared cameras for our output of the chip. Yeah.
So Gaetano is here from Hamamatsu, and he's going to give you the cameras that you need, but I want to stay in the light source. You're working on telecom wavelengths. I understand why, and everybody understands why, but Benjamin, is telecom wavelength the best wavelength for biosensing?
It depends on your application. So since we are doing spectroscopy sensing ourselves, telecom's not sufficient for the applications we need. We need the longer wavelength sources.
And everybody knows why I'm asking that. I'm going to go now to Laura. Laura, is telecom wavelength the best wavelength for biosensing?
No, no. For biosensing, I always recommend to go for the visible and visible range. And we have been working in visible range during all my life. And I'm convinced that it's the best one. That's the reason why most of us are working with silicon nitride technology, because then you can chip to visible range. And the main reason is because all the biomolecules or the bio stuff that we are using are transparent in this wetland region. So also you have then no problem with the water. Remember that we are always using liquid sample, so you have also some problem with the water absorption. So my recommendation is to move to the visible range. And also because, remember, in photonics, one of the most successful biosensors is the Surfix plus non-resonant biosensor working in the visible range. So my advice is also just to make a benchmark and to compare your sensitivity with the sensitivity of the SPR. That's the reason why it's always to work in the visible. So visible range for biosensors.
Tommy from Modulite, you see that we hear from Rockley Photonics, and they say we could go to longer wavelengths, then we can go to Laura Lechuga, one of my biggest experts. He goes to the visible. There is lots of applications here. There's going to be one more. And you're going to be really amazed at what's coming.
For biosensing, I mean, we are working for spectroscopy and so on. It's different. Then you can go to longer wavelengths, but for biosensing has to be visible. This is my advice.
Laura, what do you think of quantum cascade lasers for biosensing?
Well, I mean, this is also, remember also that for biosensing, we want to make point of care biosensing, so we have to go for very low cost component. So even if you can't solve, you're working invisible, you can solve just with a standard diode laser, so why to go for a complicated one? And also remember how difficult it is, I mean, if there is any reason to integrate all the components on chip, this is another approach. Many people is trying to integrate everything on chip. Laser, also with cascade laser, photodetector, everything on chip. But then remember, in clinical analysis, you have to make one analysis and you have to throw away the complete biochip. So there is no reason why to include all the components on chip. This is also, I think you have to make a standalone device and then just to make a cartridge where you can make a disposable cartridge. Remember that biosensor can be reused. This is another question.
Laura, you have made the perfect introduction, introduction to our final speaker today. We go to Berlin, we go to Germany, and right now in Germany, I think you guys know that. And we go to Germany and we meet a company that is trying to do the challenge that Laura just said, reducing the cost of the QCL and at the same time producing QCL-based glucose monitoring. Werner Mantele, thank you very much for presenting Optica Corporate member Diamond Tech to the entire community and tell us how we can help you do things even greater than you already are. The floor and the attention of everyone goes to Diamond Tech.
Can you see my presentation?
Crystal clear.
Crystal clear. Okay, hello everyone. Thanks for the introduction, Jose. Diamond Tech stands for Diabetes Monitoring Technology, and that is the aim of our company that I co-founded and that I am the Chief Scientific Officer, is to find a way to non-invasively measure blood glucose accurately enough to help diabetes patients. The problem is all known to you. Diabetes is a real pandemic. It's increasing right now. We count about 530 million people living with diabetes, and that is going up quite a bit. The question is, if we want to work non-invasively, we have to find body fluids that are reasonable proxies for blood. And the proxy that we have found out, not only we, but the proxy that's ideal for an optical non-invasive glucose measurement is interstitial fluid in skin, the liquid surrounding cells in skin and muscle. It's a relatively simple matrix: water, ions, albumin, glucose, phosphate, so less complicated than blood. And you find it in layers as deep as like 20 to 100 micrometers. And that is about the depth a mid-infrared laser beam can penetrate, because our indicator for glucose is a glucose signature in the mid-infrared. So in contrast to the spectral region presented by Rockley at the beginning, we are now moving deep into the infrared in the range between about 8.5 and 11 microns. And glucose has there a real characteristic signature, which is a combination of vibrations of the carbon-oxygen stretching and the oxygen-hydrogen bending modes. This is very characteristic. It's also actually, it's the vibration that our colleague from Raman spectroscopy also uses. And we use quantum cascade lasers to address these spectral range. Basically, the principle is shown on the right side. We use an internal reflection element. We use a quantum cascade laser, either tunable, broadly tunable, or with individual wavelengths as an array. We send the quantum cascade laser beam in a pulsed mode right into skin. In skin, it excites vibrational levels, basically from vibrational level 0 to vibrational level 1. You have an immediate excitation, takes about 10 to the minus 12 seconds to go down again. And there is a small amount of heat released according to the amount of glucose that has been has absorbed that radiation. And that small amount of heat migrates to the surface, enters our internal reflection element, and forms a thermal lens, a temporary thermal lens. And we send a second laser beam in this thermal lens. It is deflected, and the deflection is detected with position-sensitive diode here. That principle is called photothermal detection. We developed it from the very beginning. It's nicely patented, and it is sensitive enough to detect even small amounts of glucose. Now, the principle has been evaluated on optical benches. We built an instrument on the basis of that about 4 years ago. It's called D-Base. It's a multi-user device. It contains an external cavity tunable quantum cascade laser. It's CE certified, and we have performed a clinical validation with it on the right side. We have about 100 patients, 31 with diabetes, mostly type 1 and type, not so many type 2, more type 1. We had about 60 healthy volunteers. It was approved by Ethics Commission. Age span was up to 70 plus, male, female, approximately balanced. And we did complete oral glucose tolerance tests with each of these volunteers, meaning that over 2.5 to 3 hours, we recorded in parallel, invasively, non-invasively, including a glucose dose, and got this correlation diagram on the right side, which is consists of about almost 2,000 data pairs, and 99% and more are in zones A and B, and this is clinically accepted and is about the precision that is normally available for minimally invasive instruments. Now, from there, we have moved on. We have developed a miniaturized version of that. It's the same technology as the D-Base. It has discrete mid-infrared wavelengths in the glucose range. It has a mini glucose, mini QL array and a micro lens. And the measuring time is approximately 10 seconds, and the battery should allow about 50 measurements. It works autonomous, but can connect to smartphone and to app developers. I'm proud to show you a prototype from as of end of last year. This is about smartphone size. It has the measuring crystal here, and the complete electronics and software is working already. We are now finalizing it because it needs more wavelengths. Right now it has a limited number of wavelengths, and the precision can be higher. We expect that the finalization will be complete at the end of the year, and after the usual regulatory procedure and some more clinical tests, we expect to go to the market next summer, maybe. We have visions beyond that, because the deep pocket, as we call it, is not the end of the story, because we think that we can further miniaturize this with the help of partners and go to a wearable device, a wristband, which continuously measures blood glucose with quantum cascade laser arrays and has data logging and all the things that the diabetes patient needs as a real companion of the diabetes patient. Now, let's sum up again. We have high specificity for glucose because we use the mid-infrared. This is the glucose fingerprint. We have high sensitivity because we have a pulsed QCL excitation and our own photochemical detection. There are no alternative pathways for de-excitation after vibrational excitation. It's just heat, and it's an easy non-invasive access to glucose in interstitial fluid of skin. little spectral overlap with other molecules, and low sensitivity for ambient temperature because we record only the laser-induced delta T, and we don't have interference with pigments in skin. This may be a big difference to Raman spectroscopy because the pigments are located in deeper layers. So that's about my story, and that's at the end, this is the team in Berlin and the former team in Frankfurt. Thank you.
Thank you very much, Werner. Thank you very much for that beautiful prototype. Show it to the world, and next summer, that's just crazy. So are there any questions for Werner? Because there's a question everybody has in mind. To what extent is the price of the QCL a limiting factor?
The price of the QCL is a factor as long as you produce 10 or 100 pieces. But our laser partner, the one that develops the array with us, who is Nano Plus in this list here, has calculated that once they produce about 10,000 arrays, the array price goes down far below $1,000 per piece. And once they produce 100,000 or more per year, then the price drops to about $100, $150. So the entire cost of goods for the device can be, for reasonable numbers, can be below 1,000 euro or dollars. And that's something that the insurances are going to cover for the diabetes patient.
I was at the Wageningen Awards last Friday, and we saw that Quantium, also one company in the QCL, also with that pitch, they won one recognition. We all need this. We all need low-cost QCL. So this is something that we all need to work together to create the volume production. Werner, once again, congratulations on everything that you are achieving, and we are here to help you. I would like to close this meeting with a dear friend of mine giving the final remarks. I would like to go to Richard Crocombe. I think many of you know him. This is one of the biggest consultants in the spectroscopy domain. Richard, 2 fantastic hours. Send us away the way that we deserve.
Okay, thank you very much, Jose, for this brief opportunity. And this has been a fantastic session. My background, as you said, is in miniature portable handheld spectrometers, and I've been watching them getting smaller and smaller, and especially with the promise of photonic integrated circuits. I think I'd have just a couple of messages to the audience that you really need to understand the analytical spectroscopy. People have messed up badly, not understanding, if you like, sample presentation to the spectrometer, and then also throwing all the data into a model, getting a correlation, and it turns out to be an accidental correlation. So my message is you really need an analytical spectroscopist on staff who understand all these things, because people have messed up fairly badly in the past, not doing that. But yeah, thank you for the opportunity, Jose.
Thank you for the opportunity of having you in the room, and thank you everyone. As you know, Optica is working really hard on connecting the industry. We are growing very fast in our membership. Thank you, our latest members, Ix and Tech from Aachen, to join us actually today. We really want to create a global industry platform, and we cannot do this without having your attitude, the attitude of coming to a meeting and do your best to find ways of working together. Today we saw, for example, how Laura and Luke really had the different views, and at the end, the conclusion was we are stronger when we cooperate, and that's what we want to achieve. Here, I would like to thank all the people who made this meeting possible, and thank all our corporate members for making my dream a reality. Bye.